On the one hand, use of soil biological tests is fairly new. On the other, growers have been making calculated guesses about their field needs since, well, the dawn of agriculture. Whether their evidence was from the look of plants year after year or a sacrifice to the gods that was deemed subpar, we just have a bit more accuracy and quicker turnaround now. Although, real talk, some of it may still be Zeus's fault. There is always a cost to technology, but the tech has advanced, the costs have lowered, and these tests are becoming more affordable and available. Plus, I know a guy. I can get you a test by like 2 o'clock. So interest and availability have increased, but in terms of efficiencies, some kinks are still getting worked out. There are still wait times of 4 to 6 weeks to receive data back from certain tests. Ideally, farmers can combine both a conventional chemistry approach and a biological approach to provide a more holistic picture of what is happening in the soil. This means testing for both available chemistry and the biology that is making it plant available. Let's look at some tests. The first is the Haney Test. The Haney Test measures different forms of nitrogen, like nitrate, ammonium, and organic, which is nitrogen in the amino acid form. Testing all 3 forms provides a broader picture of the total nitrogen that is available to soil microbes, rather than boring nitrate nitrogen alone. Just kidding, nitrate nitrogen, we love you. Text me, call me. Nitrogen in the ammonium or amino acid forms is more stable, long-term, and stored in soil organic matter, and is made plant available through microbial activity for plant uptake. It's like the cool mom of nitrogen. It has the best snacks. Next, we have PLFA, phospholipid fatty acid test. This is the awkward 80s presidential fitness test for soil life, kind of like a measurement of the fat that is extracted from microbial species. Ooh. The fat is then categorized into buckets of species such as bacteria, protozoa, and fungi, which shows the total amount of these microbes that a soil contains, but that's it as far as the test goes. A general rule is the more diverse your soil, the healthier your soil, a lesson that a whole lot of human organizations are still working on too. To identify specific microbial species, a third option is genomics, or DNA sequencing. This technology has been around for over a decade, but only has become popular recently. Different labs use different methods. For example, a sequencing technique termed metagenomics, or other forms of DNA sequencing that work toward the same result of microbial identification, like your first day at a new high school. One big benefit of DNA sequencing is that you get to understand the presence of specific groups within your soil. Some of the functions and associated cliques, if you will, you might see reported include nutrient availability, phytohormone production, stress adaptation, and biocontrol. And finally, there is microscopy. This methodology uses a microscope to identify species of microbes. As you can imagine, it's time-intensive because a live human is examining one microscope slide at a time. This requires investment of equipment, training, and hours of repetition and practice to identify particular species, so the viewer actually knows what they are looking at. Generally, microscopy will easily confirm that most soils are bacterially dominant, so finding your fungal and protozoan friends may take additional time. It's important to remember that, much like Congress, the lab results may complain about what's wrong without offering solutions to fix it. Soil health testing is in its infancy for gaining popularity and use by farmers and agronomists. While these tests are being used now to inform management decisions, more research by university extension and the scientific community is needed to support their interpretation, because the main goal is to have more good stuff and less bad stuff. And for that, we still rely on general recommendations regarding 6 principles of soil health. Cover the soil, minimize soil disturbance, increase diversity, maintain continuous living cover, integrate livestock, and know your context. As more research becomes available, the ability to interpret and use soil biological testing more confidently will increase. We are really beginning to understand how it's biology that makes the chemistry in the soil available. And these are exciting times to be fungi, or protozoa, or even bacteria, nematodes, or water bears, or actinomycetes. Actinomycetes? 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 Actinomycetes?